Hi guys, this is Malinki. Welcome back to my channel, Voice of Malinki. Today I will discuss about Immunology Lecture 9, differences between cytokine, hormones and growth factors. And if you are new in my channel, please subscribe my channel, and if you like my video, please do like, comment and share my video. Okay, so first we should discuss their similarity. So all these three, all hormones, cytokines and growth factors, all are actually chemical messengers. So that means they mediate intercellular communication and cell activity. So they mediate intercellular communications and cell activity. So cells can communicate with each other via these messengers, and cells activity depends on them. Now all three act in autocrine, paracrine and endocrine manner. So first of all is autocrine action. So they bind to receptor on the membrane of the same cell that secreted it. So suppose this is a cell, this is actually a producer cell, and this cell secretes either a hormone or cytokine or growth factor, whatever it is secreting this, and these secretion, this secretory product will act on this cell only. So in this case, this cell is producer cell and also target cell. So the substance, chemical substance that is secreted by this cell, it will act on this cell only. Now next is paracrine action. So paracrine action, actually in this case they bind to receptor on a target cell in close proximity to the producer cell. So here what we will see, that suppose this is the producer cell and this is releasing, or this is secreting these chemicals, and they will actually bind another cell. So this is the target cell, and they will bind this cell and will act on this cell, and this cell is actually very close to this producer cell. So this is the paracrine action. Now endocrine action. So endocrine activity is like that they travel through circulation and acting on target cells in distant parts of the body. So suppose this is the producer cell, and some substances are getting released from these cells. Now these substances will move to another cell, that is the target cell, and this cell is away from these producer cells. So these substances will actually move, they will move through the bloodstream, and then they will reach this target cell, and then they will act on this. So this is the endocrine action. So cytokine, growth factor, hormones, they all can actually act either autocrine or paracrine or endocrine manner. Now one more similarity is there, that all of them, they regulate the gene expression. So all of them regulate the gene expression in the nucleus, and ultimately they stimulate a biological response. So in the cell, I already said that they act on a target cell. So what they will do, actually they first of all they bind the target cell, it's okay, then they will enter that cell, and then ultimately they will act on the nucleus, and they regulate the gene expression. So they regulate the gene expression, and according to the gene expression this target cell will act. So these are the similarities. So they, all of them are doing the same thing. Then what are the differences? Okay, so first of all, difference between all of these, that is growth factor, cytokine and hormone. So cytokine and hormone are types of growth factors. So cytokine and hormone, they are actually growth factors. So growth factors might be of different types. Among them, cytokine is one type and hormone is another type. So first thing you need to understand that both cytokines and hormones are growth factors. So they both are actually growth factors. Although apart from cytokine and hormone, growth factor includes many other substances. Now, if you ask me an example, suppose this is the epidermal growth factor, epidermal growth factor, that is EGF. This epidermal growth factor is one of them. It is actually a growth factor. This is growth factor only. But it is neither a cytokine nor a hormone. But it is a growth factor. So like that, growth factor includes many more substances. So among them, cytokine and hormone are two of them. Okay, so now we have to know difference between cytokines and hormones. So cytokines and hormones both require a cell surface receptor to act on that particular cell that we already know. Now hormone is of two types. We know hormone is of two types, right? Hormone, they are either peptide hormone or steroid hormone. Right? Now steroid hormone, actually they don't require any cell surface receptor to enter a cell. Why? Because steroid hormones are made up of lipid, right? Steroid hormone are lipid only. And hence they are lipid soluble. Because they are lipids, so they are lipid soluble. So as they are lipid soluble, they can easily cross the lipid bilayer membrane of a cell. So this is the plasma membrane, that is lipid bilayer membrane. We all know that cell membrane is made up of lipid bilayer. So these steroid hormone actually can easily cross this lipid bilayer of cell, and they also can enter, or they also can cross the nucleus. This is the nucleus, so they also can cross the nuclear membrane also. So for steroid hormone, it's not a big deal to enter into nucleus. They don't need any extra cell surface receptor for their entry into the cell as well as nucleus. So they directly enter a nucleus. So it's not a problem for them. But cytokines and peptide hormones are proteins, right? And they are not easily permeable in the cell cytoplasm and nucleus. They require a gate to enter, and cell surface receptors are those gates. But that gate is different for peptide hormone and cytokine. So that gate is different for peptide hormone and cytokine. That gate is not similar, that gate is not identical for them. So one thing you need to understand here, that cytokines and hormones both act on gene transcription finally, right? So because cytokine and hormones, what they will do ultimately in the nucleus, they will ultimately help in gene transcription, and neither of these two molecules are allowed to enter the nucleus. Therefore they need some molecules who will convey the message in nucleus, because cytokine and hormone, they can now enter the cytoplasm via some receptors or cell surface protein, you can say. So they are now in cytoplasm, but they are now in cytoplasm, it doesn't mean that they can enter nucleus also, because nucleus is very much selective. It doesn't contain any gate to allow them. Now what so they have to do something, right? So what are those molecules that are allowed to enter nucleus? So those molecules that are allowed to enter nucleus are actually three molecules. So you need to understand these things very well. So first one is pKa, that is pKa is protein kinase A, right? Next is pKc, that is protein kinase C, and one more is PTK, that is protein tyrosine kinase. So they are all kinases. So from their name we can understand they all are kinases. And what they do, they catalyze the phosphorylation of other proteins within the cell, because kinases do the thing that they phosphorylate some other proteins. So all of them, they will actually phosphorylate some proteins. So they will phosphorylate some proteins, right? Now, enzymes that are activated or inhibited by phosphorylation actually enter DNA and regulate gene expression to alter cellular activities. So, suppose this is one enzyme. Now, this enzyme is phosphorylated by any of them, right? Now, phosphorylation can make a enzyme either activated or inhibited, right? So depending on that enzyme will act. Now both cytokines and peptide hormones actually activate them, but this activation mechanism is different. So cytokine and peptide hormone actually activate these pKa or pKc or PTK. So they are the ultimate molecules, they are getting activated by cytokine and hormone. But what is the difference? This activation mechanism is different. So we will study that mechanism. Then we can understand the difference. So for cytokines, cell provides two types of gates. First one is receptor. First one is this one. Okay. So this is the receptor that is linked. This is the receptor and that is linked to PLC. PLC. PLC is phospholipase C through a G protein. Here you can see this is the G protein. So this is one gate. This is cytokine receptor. You can say it gate 1. You can say it gate 1. So this gate 1 is a cytokine receptor. Mind it, this is cytokine receptor. And what they do, this gate is actually linked to phospholipase C via G protein. So this is gate 1. Now when a cytokine binds this gate 1, so a cytokine now binds this gate 1. Now this PLC will be activated now. Okay, whenever the cytokine binds this receptor or gate 1, this PLC will be activated. So activation of PLC, what it will do? It will cleave this PIP2. PIP2. It will cleave this PIP2. PIP2 is, what is PIP2? It is phosphatidyl inositol 4,5-bisphosphate. So it will cleave this PIP2 and what will it form? It will form DAG. This DAG and IP3. So DAG is, what is DAG? DAG is diacylglycerol and IP3, that is inositol 1,4,5 triphosphate. So what it will do? So activation of PLC, now it is cleaving PIP2 to form DAG and IP3. So DAG and IP3 are now formed. Now IP3, so they will do different things. First of all, IP3 binds IP3 receptor on endoplasmic reticulum. So this is the endoplasmic reticulum and it possesses some receptors, IP3 receptors it have. So these IP3 will now bind this IP3 receptor. These binding, that is IP3 and IP3 receptor binding on endoplasmic reticulum, it will stimulate endoplasmic reticulum to release calcium. Because we know that endoplasmic reticulum stores calcium. Now this binding helps this ER to release calcium. So now free calcium are released in the cytosol. Now this free calcium, what it will do? It will now bind this calmodulin. This calcium will now bind calmodulin. So this calcium calmodulin complex that is now ready. It will activate protein kinase A. It will activate this protein kinase A in the presence of cyclic AMP. So in the presence of cyclic AMP, calcium calmodulin complex will actually activate pKa. So this is one thing. So from here you can understand that pKa is ready. So we know that pKa is essential for the nuclear function. So pKa is now ready. And I have already told that DAG is one more thing. So what DAG will do? DAG will do some other thing. That DAG will bind to an activate protein kinase C, PKC. So DAG will actually activate this PKC, right? So now PKC is one more. So PKC is now also made. So pKa is made, PKC is made. So these two are required to do some function, function that can actually help in gene expression. So pKa and PKC are ready. Okay, so now coming to gate 2. One more gate is there, that is gate 2. So gate 2 is, this is the actually gate 2. Gate 2 for what? Gate 2 for some other cytokines. So this gate is also called protein tyrosine kinase, PTK. This gate has another name, that is PTK or protein tyrosine kinase. So this is one more receptor that is found in the cell surface that also can bind cytokines. So its role is very much direct. So for like PTK is made, because here I have already told that PTK is required. PTK is required because PTK is a kinase. So that we require PTK in the cell. So here PTK is made, pKa is made, PKC are made. So they all are made, and what they will do, that I have already told, that they will actually phosphorylate some enzymes, and those enzymes will enter nucleus and they will do their function. They will help gene expression like that. Okay, then these are all about the cytokines, how they are functioning. Now next is hormone. So for hormone, this gate is different. So this gate is different means, suppose this is the gate, this is the gate, this gate is for hormone. So here peptide hormones can bind this receptor, right? So this gate is also linked to AC. This is AC. This gate is linked to AC, that is adenylate cyclase. AC is adenylate cyclase, and this gate is linked to AC through G proteins. So this gate is linked through G proteins. Now when hormone binds this gate, so when hormone binds these gate, so what happens? This AC gets activated and it increases CAMP level. So this AC will increase CAMP level. Now CAMP in the presence of the calcium calmodulin complex. So calcium calmodulin complex are there in the cell. So CAMP in the presence of calcium calmodulin complex, they will activate pKa. So they will activate again pKa. So these peptide hormones actually activate pKa. So again pKa is ready. So one more difference. So this is the difference of their mechanism. So how they actually work. The ultimate thing is same. They all actually work on nucleus. They actually work to express some gene, but the mechanism is little bit different. The intermediate substances that are working here, these are little bit different for hormone and cytokines. And one more thing, one more difference you can find so cytokine, between cytokine and hormone. So classic hormone, actually hormones circulate in nanomolar concentration. So you can get hormones in nanomolar concentration. Nanomolar, so hormones in nanomolar concentration means 10 to the power minus 9 molar. So in this concentration, hormones are actually found in the blood, and in the cytokine, for cytokine. So this concentration is 10 to the power 12 molar, or you can say it as picomolar. So it is picomolar. So cytokine, very little concentration of cytokine you can find. So hormone, the concentration of hormone is greater than concentration of cytokine. So this is the difference, and hope you've enjoyed this lecture, and stay with me for the next exciting lecture. Bye bye.